So today is Monday, and every period we are going to cover on Wednesday. You're only going to be here for a shortened period of time. You're going to have your regular schedule, but you'll be out by 12. I don't remember how long the class is, but every period I'm going to cover a different topic. I'm going to make one recording, and come Wednesday afternoon, after 12, I'm going to upload the review video. By 12 o'clock, you will have an entire review for the test. So it is what it is. It's an 80-minute exam, so you can expect a lot of questions. It's been a month and a half. It's a midterm, so you can expect I'm covering everything, including and especially every lab. I mean, that should be clear. But if it's not, I'm making it clear now. Now, Monday. Today, as you see in the syllabus, the weekly syllabus is carbohydrates and lipids. So today we're going to talk about carbohydrates. Let's start with that. A lot of you have heard of it. You've seen it you've seen carbohydrates in your nutritional breakdown. You're worried about carbs. You know them as carbs. It's a shorthand for carbohydrate. So you tell me, what do you know about carbs? Does anybody know anything about carbs? Yeah. Sugars, yeah. What'd you say?
Kind of fatty.
Kind of fatty. No, they're not fatty, but they add fat to you. They can make you fatter. So if that's what you mean.
Yeah.
So carbohydrates are sugars. Sugars are carbohydrates, rather, I should say. And they have a general formula, as I've mentioned before, CHO or CHO. For every one carbon, you expect to find 2 hydrogens and 1 oxygen. That's because every carbon, or almost every carbon, has, when you look at some of the simpler carbohydrates, let's look at glyceraldehyde. This is one of the more important intermediate molecules. And we're going to use glyceraldehyde later in a minute when we talk about fats. Now, if you count these, you got 1, 2, 3 carbons. So how many hydrogens would you expect with 3 carbons? 3 carbons, what would you expect? 3 if this were a 3, and for every 1 you get 2 of these, you'd expect 6 hydrogens. Let's count them. 1, 2, 3, 4, 5, 6. If you have 3 carbons, how many oxygens would you expect? 3. 1, 2, 3. So again, it fits. Not every carbon has 2 hydrogens and an oxygen, but it turns out that when you have a sugar, any kind of carbohydrate, this is the form it takes. And by the way, I want to remind you again, you should be checking your Schoology messages and your Jupyter messages. You don't have to do it over the weekend. You can check on Friday night. You can check Monday morning. What's best for you is Friday night. So this is known as glyceraldehyde. Now I'm going to add to your functional groups. I'm going to add to your functional groups, and there's a word here that you should become familiar with. You're probably not going to see it on the test, but it's still an important word. It's this word aldehyde. And the aldehyde comes from this, having this double bond oxygen at the end. You see, that's not a carboxylic acid. Do you understand that? A carboxylic acid, remember that functional group, this could be part of your review, by the way, because you will have to be able to identify it. C double bond O, OH. That is a carboxylic acid. That's actually probably a better way of drawing it would be C double bond O, OH. That's a carboxylic acid. You should know that. It's part of an amino acid. It's one of your functional groups, et cetera, et cetera. Notice that this is not that. There's no OH on this. This is called an aldehyde. So this is glyceraldehyde. Another type of aldehyde sugar, or sugar that has an aldehyde in it, is a carbon of 1, 2, 3, 4, 5. You know this sugar. You should know it. What sugar is this? 5. The one sugar that you've been introduced to. Say again? Ribose, correct. So here you have a ribose sugar. It looks different, doesn't it? You agree it looks different? Okay, we're gonna address that difference in just a second. What do you used to see? How are you used to seeing it? That's right. It looks like a pentagon. That's good. That's good. That's a ribose. Now, ribose also has this aldehyde group on it. We don't call it aldehyde. We just call it ribose. But it is one of those that has this aldehyde group on it. It's a ribose. There's two types of sugars. Those that are aldehydes and those that have another group that we'll introduce in just a second. Now you have another sugar that has 1, 2, 3, 4, 5, 6. Six sugar. If I told you that this is a sugar that you know, what sugar could you name it? Oh, good guess. Except the deoxyribose has 5 carbons just like ribose. So good guess, because you're thinking deoxyribose, ribose, that's what we covered. So that's really, really important that you do remember deoxyribose and ribose. But what you need to point out is that ribose has 5 carbons. This has 6 carbons. It's a big difference. They alternate. One goes up, or if they were a ring, one would go up, one would come down. What's another sugar you know? It's not deoxyribose. No, that's not, DNA is not a sugar, it's a nucleic acid. What is it? Lactose is a good guess. It's not. Glucose is correct. So those are the sugars you know, and there's many other sugars, but those are the ones you know. So it's important that, do you have to recognize these? No, you do not. I'm not going to ask you to look at these and recognize them. I'm only drawing them to point out the differences. So I'm not going to have you look at this and say, wow, is that a glucose, or is that a ribose, or is that a lactose, or is that a glyceraldehyde? No. Okay, so this is not one of the things you have to memorize. Then when we look at this one here, what do you notice is a difference here? And you know this sugar as well. And this thing, the fact where the double bond is the big difference. And that difference actually makes it more difficult for you to digest. It's more difficult for you to process this sugar. And you drink it all the time. You drink it and eat it all the time, but mostly drink it. Lactose is a good guess, but it is not lactate or lactose. So, and you're right, it is difficult for you to digest lactate because we don't have the enzyme for it. But the fact that this sugar has this group here in the middle, you see the difference? So this particular group has an OH double bond O in the middle of the group, not at the end. So these are all aldehydes. This one is different. This is what we call a ketone. Has a ketone group in it. So what do you think is going on there? What kind of sugar do you think? It's a twin to glucose. These two are twins, except this twin has a double bond O. It's notice 1, 2, 3, 4, 5, 6. There's 6 carbons in both. They're both hexoses. This is a pentose. Yeah. Is it what? It's very similar to glucose. Very similar to glucose. You call it fructose. Fructose is a ketone, and because it has this carbon in the double bond O in the center here, or in the middle instead of at the end, this is known as a ketone. Much more difficult for you to digest. That's why drinking soda pop that has high fructose corn syrup, that means it's high concentrations of fructose, it's very difficult for you to digest it. So it actually puts a lot of pressure, a lot of stress on your liver. So and a lot of people think that drinking the high fructose corn syrup in Coca-Cola, in Pepsi, etc., can cause some serious issues long term. So before you get your next 3-gallon drink from McDonald's, consider not eating high fructose corn syrup. Consider reducing the amount that you drink. So those are the main sugars, and it turns out that, like we said, when we draw ribose, all of these can form rings, except glyceraldehyde. The ribose, glucose, and fructose can all form rings. Ribose makes a ring that looks like this. It's 5 carbons, as you know, 2, 3, 4, and 5. And we call them prime. Glucose makes a ring that looks like this. Glucose looks like this, and it has 1, 2, 3, 4, 5, and 6. Oops, I put that's why I messed that up. Sorry, I always mess that up. The oxygen goes here. 1, 2, 3, 4, 5, and 6. Again, they're primes. So when you're looking at the linear form, we tend to draw glucose. And in the book, in the chapter reading, and in class, we'll draw just the hexagon. And most of the time, we'll be talking about glucose, because glucose is the primary energy source for your metabolism of your cells. This is what plants make for us. This is how we take apart. This is what gives us energy. Ultimately, it'll be glucose, and we talk about cellular respiration. The ribose, and remember its cousin, the deoxyribose. Ribose and deoxyribose, they're used in DNA and RNA. All right, so don't confuse that. So the difference between aldehydes and ketones are significant. Now these are all monomers. Meaning what? What does that mean, that these are all monomers? We call these monomers, by the way, monosaccharides. That's right, single building blocks. In this case, single building blocks of sugar. Saccharide means sugar. So these are single building blocks, correct. So they're Single building blocks, what does that mean to you? What are you going to do with them? What do you do with single building blocks? You put them together. We make polymers, right? Ribose and deoxyribose, we've looked at the polymers, haven't we? You built them, haven't you? I mean, you had other parts to it. You had phosphate and you had a nitrogenous base. But we made, ribose is part of a monomer that we call a nucleotide. That you have to know for the test. I'm telling you, because you want to know, you want a study guide, I'm giving it to you right now. I'm telling you that will be on the test. What is a nucleotide? What are the parts of a nucleotide? That question was in the lab. That question was asked in class and given in class. And now I'm telling you now, what is a nucleotide? What are the types, what are the parts of a nucleotide? Again, if you want to review, go back in your notes or go back in your in the video, you can. The three parts of a nucleotide are ribose or deoxyribose, depending if it's RNA or DNA, a phosphate, and a nitrogenous base. Remember the A, the T, the G, or the C. Yeah. I already wrote it down. It's in the previous video. I don't want to cover it now because I want to focus on carbohydrates. But I will go over it again during the review. What I would do is I would go back and look in the book, go back and look at the video, go back, study groups, comparing each other's notes. The stuff I've been telling you to do all month, for 2 months now, right? Get together with your friends, or colleagues, come and see me during lunch. I'll be here during 4th and 6th. I'll be covering 2 classes. You can come and see me during 4th and 5th, at 6th if you get a pass from your teacher. Whatever you need, I'm here for you. All right. So when we're looking at sugars also form polymers, very important polymers we're going to talk about right now. You're sitting on one of them. Did you know your stools are made of sugar? Did you know wood was made of sugar? Yeah, it's made of sugar. So let's talk about that. There's two, we call these polymers, a polymer, remember, is this taking a unit and building something. We call these polysaccharides. Where saccharide again means sugar and poly means more than one. Now one of them I know you love. I don't know a single kid that doesn't love it. One polysaccharide you love very much, and that's called starch. I know you love it because all of you eat French fries and potatoes. And potatoes are starch. They're made of starch. Or they store starch. I shouldn't say they're made of it. And what they are is they just simply are sugar, carbohydrates, that are linked together. They're just linked together over and over and over again in a long chain. And we call that long chain starch. And if we took this, oh man, I hate when that happens. If we took this single glucose molecule and had a bunch of them, and we connected them in a long chain, we would have something called starch. And starch has it's very long. They get connected like this, and they're very long strands over. They're not quite, the geometry is not quite right. Did you get that, Carly? The geometry is not quite right.
What?
Did you get what I said, that the geometry is not quite right? Did you get that? No? Okay. So, anyways, the starch is less branched, right? It's for storage. What do you need to know for the test? You do not need to know the structure. You need to know that starch is used by plants to store sugar. Plants make sugar for themselves. Let's be clear on that. That's an important point. Plants make sugar for themselves. We steal them. We steal the sugar. So when we eat fruits and we eat potatoes, we're stealing the sugar that the plants made for themselves. They eat their own. This is their food. They made their own food. That's an important point. So plants have another, now this is for storage, energy storage, right? Energy storage. There's another type of polymer or polysaccharide known as cellulose. That's very branched, and they have different, and the difference is that cellulose and starch have different linkages. They have different ways of combining the monomers. And because of that, you cannot digest cellulose. It's made by plants. Again, plants for structure. Even plants can't eat this. Once plants make cellulose, they can't eat it. This is their skeleton. You're sitting on the skeleton of a tree. They make the skeleton from sugar. This is just sugar, just the same sugar you eat, glucose, except they're all put together in a way that we can't break down. Yeah.
You said cellulose is a monosaccharide?
Is it what?
A monosaccharide?
Monosaccharide. No. Cellulose is a polysaccharide made of glucose. So these are all polysaccharides. Polysaccharides. They're made, but it's a good question. They're made of glucose. Both starch and cellulose are made of glucose, but they're connected differently in such a way as very few living things can break it apart. Specifically, fungi, and some bacteria. Those are the only things that can break up cellulose. You can't eat cellulose. I can't eat cellulose. Cows can't eat cellulose. But why do cows eat grass then? Grass is made of cellulose. Why do they eat grass if they can't eat the cellulose? Well, it turns out they can. So can termites. But they can't eat it themselves. They don't have the enzyme to break it up. They use bacteria in their intestines to break up the cellulose. Just like you have bacteria in your intestines to help you digest, they have bacteria in their intestines that help them digest. So termites and cows, they can't digest your chairs any more than I could. But if they have the bacteria in their gut that allows them to break it up, then they can break up the cellulose. That's the special enzymes. These organisms have a special enzyme that helps them digest cellulose. If you had the enzyme, you could eat this the way you eat cotton candy. That wood that you're sitting on. It would taste just as sweet. It's just glucose. It's literally the same stuff that you have in any candy that you eat. The difference is how you connect them. You see how amazing this one little change? They just change one thing. Instead of doing this, connecting them this way, they connect them that way. Just like your Lego, just like your Minecraft. And just changing that makes the difference between food, starch, right? Food, and structure. As you know, it's structure because you're sitting on it. We make tables out of it. We build our walls out of it. So it really is structure. So that's it for plants. Now animals have a couple ways. You have a way of storing glucose as well. One way you have of storing glucose, that's plants. Animals, including humans, use glycogen. And humans store it, again, it's a polysaccharide, a bunch of sugar molecules connected together, and yet one more way that, and this glycogen in mammals, it's stored in liver. So in your liver, you put a bunch of glycogen. You store anytime you eat extra sugar, if you have extra sugar, your body takes that sugar and puts it away in your liver. When your liver gets full, you start to make fat. So when you're looking at mammals, we store glycogen. So again, it's a polymer, it's a polysaccharide of glucose, and it's found in humans. It's also in humans. Humans are mammals. That should not be a surprise. And we call that glycogen. There are a few other polysaccharides, like chitin. Chitin is found in insects. It's a structural. This is structural. So chitin is found in insects. It's structural. You know that. Have you ever stepped on an ant and felt a crunch? That crunch is their exoskeleton. I think it's gross, the poor little thing that was just alive and now you just killed it. But that exoskeleton is made of sugar. It's glucose. It's just put together in a different way. So by putting these monomers together in different ways, we can do different things. And those are the 4 that you need to know for your test. So if you know that much, which one's animal, which one's a plant polysaccharide, which one's an animal polysaccharide, which one you can eat, which one's used for structure, which one you can eat, which one's used for structure. We don't have a structural polysaccharide. We don't make chitin for ourselves. We don't have an exoskeleton. But a lot of exoskeletons are made of chitin. Are we good then? So you have to know those 4 for polysaccharides. You have to know those sugars for monosaccharides. You don't have to know what they look like. You just have to know that they're monosaccharides. You don't even have to know which ones are ketones and which ones are aldehydes, just to be clear. You just need to know that they're monosaccharides, and these are polysaccharides, and these are the 4. Are we good? All right, so now let's take a look at lipids. Is it? So what time are we leaving? So no, we have enough time to finish. So lipids, they can be all kinds of lipids, but we have this molecule that's related to glyceraldehyde called glycerol. Again, it's three carbons. And fats, we should talk about what we're talking about lipids here. You have to know lipids in general. Lipids are fats. So you know what fats are. You eat them. If you wash dishes, you see them floating in the water. Do you not wash dishes? You know, that greasiness is fat. When you see it in the chicken, when your mom's cutting up chicken, you see that white part underneath the skin. That's fat. You have fat. It's part of nature. It's how we store long-term storage of energy. And what we do is we make this long chain of, anytime you see a long chain, so let's be clear, of carbons, of just carbons and hydrogens, or rather mostly carbons and hydrogens. It's a long chain. It could be up to, I don't know what the longest one is, but it's long. So you have this long chain. At the very end, they're mostly hydrogens. All these are all hydrogens. I would draw this because you will have to know this structure. The reason you have to, I don't think it's that hard. It really should just be one new thing that you have to learn. One new structure. This is it. You should already studied and know DNA, proteins. Anyways, I'm not going to keep going. This is why I keep telling you, you got to keep up. You're not over. We're only at the beginning. If you're not keeping up, it's going to get harder and harder. At the very end, you have this carboxylic acid group. Oh, why are you not working? There you go. At the very end of this is a carboxylic acid, double bond O, single bond OH. And that's why we call this a fatty acid. You've heard of fatty acids? So this is a fatty acid. These are all hydrogens. It's mostly hydrogen and carbon. So we call these hydrocarbons because it's hydrogen and carbon. Do you all agree that it's mostly carbon and hydrogen? You have very little oxygen. If you remember that, when you're looking on a test, you should be able to identify a fatty acid very quickly. A lipid has mostly carbon and hydrogen. A sugar has carbon and hydrogen and oxygen at a 1 to 2 to 1 ratio. If you know that, you're set. Yeah.
You said
a fatty acid or fat has mostly carbon and hydrogen. That's why another word for fats is hydrocarbon. You use hydrocarbons in your car. A hydrocarbon example is gasoline. It's a derivative of fossils, leftover fats. Hydrocarbon. All right? So we can take this glycerol molecule and we can make something that all of you, your parents certainly would have been tested for. We can make a triglyceride. So we can make three fats or 3 fatty acids. We can attach 3 fatty acids to this. If this is what you have, I hope you notice that there's an extra bond that we can attach. And because this is an OH, and because this has an oxygen on it, this has an OH group on it, we can take and link them together. We can link them together and take water out using dehydration synthesis, and we can connect these this chain. I'm just going to put a chain like this chain of hydrogen and carbon, and we can do that at every turn. Now, sometimes these carbons have double bonds. Do you see that? Do you see this double bond? And that double bond will give it a kink. This is very important, so make sure you get this straight. Sometimes there'll be a double bond, and because there's a double bond, the hydrocarbon will be kinked. It'll have a little turn to it. Sometimes it has more than one double bond. It could have 2 double bonds here, and then another one here. So these double bonds, let me label each fatty acid. Do you recognize that this is a fatty acid? This is a fatty. That's this drawing here, and just connecting just the carbons, just like we did with the 5-sided ring that you put together, just like we did with the 6-sided ring. We're just connecting the lines. At each connection is a carbon. We're not showing the hydrogens. This is a fatty acid. This is called a saturated fatty acid. You've heard of that, yes? Saturated fats? That saturated fat means no double bonds. Every carbon every carbon has a hydrogen. That's what we call saturated. Saturated means it's full. Here, this is an unsaturated fatty acid. Why is it unsaturated? Because you have a double bond. One double bond. What do you think we're going to call the other one? You've heard of. What do you think we're going to call the other one? Polyunsaturated. You've never heard these words?
I've heard saturated and unsaturated.
Yeah, not poly.
Well, polyunsaturated is the best fat you can have. If you're going to eat fat, and it's good for you, by the way, fat is good for you. You need it. It's required. So when people tell you eat low fat, that's still being debated whether we should be low fat or low carbs. That's up to you. Some people follow the Atkins diet. Yes? You've heard of that?
Yeah.
All right. If you haven't, you should become familiar with it. The Atkins diet is still in debate. People think there might be a little too much pressure on your kidneys. Might be bad for your long-term health. But you can follow unsaturated fatty acids. Polyunsaturated fatty acids are really good for you. Some saturated fatty acids are okay. A little bit of this is all right, but not too much. And that's basically all there is. That plus these ring structures called steroids. You've heard of steroids?
Yes.
Testosterone.
Yep.
They're giant ring structures. Testosterone is just one of them. They look something like, I'm just sketching it, but they look something like this. I mean, you see how big this is? I mean, they're giant. And this is testosterone. This is cholesterol. You've heard of cholesterol?
Yeah.
That's also a lipid. So these are lipids. This is what lipids are. That's it. They are storage forms. They could be hormones. And this is the last of it, hormones or storage, long-term storage of energy. Or they could be structural. So I can see a question where I would ask you on the test, this is a lipid, how can it be used? This is a carbohydrate, how can it be used? This is a protein, how can it be used? Each one has different sets of properties, different qualities. All right, good luck.